All right, are there any questions? So we are actually getting to the stage where we will finish our discussion of molecular biology, right, for this course. I mean, I think today I'll be able to finish that, right? And then we'll move to the engineering stuff. So last time I was talking about how you can use frog eggs, right, to test the activity of the M-phase promoting factor, right? Because the Xenopus oocyte is just at the stage where it is ready to basically move into the M phase, okay, of the first meiotic cell division, right? And so if you give it M phase promoting factor, that's going to happen, right? If you give it, enzymes from, if you give it an abstract that is from some other stage of the cell cycle, right? That does not happen. Okay. Yeah. What? Oocyte will have, yes. The chromosomes have replicated, yeah. It has not halved as yet. Okay, so the egg has not been produced as yet. Okay, so it is the egg is going to be produced from its precursor, right, by meiosis, right? So there will be DNA replication followed by 2 rounds of cell splitting, right? So this one is arrested just, the DNA replication is over. It is arrested just before the first round of cell splitting. Because that's the characteristic. So if you give it the M-phase promoting factor, it immediately splits, okay? If you give it an abstract from some other stage of the cell cycle, that doesn't happen, okay? So that's how they called it the M-phase promoting factor. So in such experiments, it was found that an abstract from an M-phase fertilized egg immediately drives the oocyte into M-phase, whereas cytoplasm from a cleaving egg at other phases of the cycle does not, right? So when this was initially discovered, they didn't know the activity, right, or the factor that was responsible, so they called it the M-phase promoting factor, right? But later on, they found out that it has got a kinase, right, and it also needs another unit that is called cyclin, right? So the kinase is the catalytic unit, and the cyclin is the regulatory unit. The cyclin has to come and bind that kinase or the cyclin-dependent kinase, right, in order to become active. And the activity varies like this. The M-phase promoting factor, the activity increases rapidly, right, at the start of M-phase, right? So it will drive cells into M-phase, and immediately after M-phase, it falls also very rapidly, right? So you can see between one M-phase to the next, you have the interphase. Now cyclin, the concentration of cyclin gradually builds up during interphase, and then it peaks during M-phase, right? And then it comes down very rapidly. Again, it gradually peaks up, right? So we'll try to explain how that's happening, right? Now, by phosphorylating key proteins, that means adding phosphate group onto key proteins, the kinase, the cyclin-dependent kinase, causes a lot of phenomena that are associated with mitosis to occur. For example, the condensation of the chromosomes. Then you want the disintegration of the nuclear envelope, right? And that happens during which phase? Do you remember? Which phase of mitosis the disintegration of the nuclear envelope happens? Prometaphase, okay. And then the formation of the mitotic spindle, right? So all these things need enzymes, and this kinase basically, makes sure that all these things happen at the right time. However, this M-phase promoting factor kinase is not capable of acting by itself, and it has to have a specific cyclin bound to it in order to function. Now, biochemical experiments using cleaving clam eggs, that means clam eggs that are dividing very fast, led to the discovery of cyclin. Cyclin was initially identified as a protein whose concentration rose gradually during interphase, right? And then fell rapidly to 0 as the cells went through M-phase. So this is like a timer, okay? I mean, it's building up, and then, maybe a capacitor getting charged, then discharged, okay? So doing the timing, right? And then it fell rapidly to 0 as the cells went through M-phase, repeating this performance in each cell cycle, as shown in the figure earlier, right? So the MPF, or the M-phase promoting factor, is a protein complex that has 2 subunits. There is a regulatory subunit, which is a cyclin, right? And then there is the catalytic subunit, that is the mitotic cyclin-dependent kinase. Now, many of these cell cycle control genes, they have been remarkably conserved during biological evolution. As a result, the human version of these genes, okay, that you and I have in our cells, okay, that'll function perfectly well if you introduce it into a yeast cell, right? Now, the manufacture of the cyclin component of this M-phase promoting factor starts immediately after cell division and it continues steadily through interphase, right? The cyclin is going to accumulate, right, so that its concentration rises gradually, and it helps time the onset of mitosis. Possibly after it crosses some threshold, that's when mitosis takes place. Its subsequent rapid decrease helps initiate the exit from mitosis. And the sudden fall in the cyclin concentration during mitosis is the result of the rapid degradation of the cyclin by the ubiquitin-dependent proteolytic system, right? You may have forgotten, probably you did not because you took the test, right? You remember this ubiquitin is a protein that is used to tag something for trashing, okay? So that's the kind of system that actually is responsible for degradation of the cyclin. Now the MPF activation, the M phase promoting factor activation, initiates a process which after some time delay leads to the ubiquitination and degradation of the cyclin, thereby turning the kinase off, right? Because the cyclin is the thing that does the timing, right? So if the cyclin is destroyed, then the kinase in the M phase promoting factor gets turned off. Now if you looked at the figure, right, you would have observed that the cyclin concentration increases gradually throughout interphase, right? Not fast, okay? It doesn't increase rapidly. However, the MPF, the M phase promoting factor kinase activity switches on abruptly at the end of interphase, okay? So the cyclin cannot be the entire story, okay? The changes in the cyclin could not be doing the entire timing, right? So the cyclic variations in the cyclin concentration alone cannot completely explain the MPF kinase activity. This is due to the fact that this kinase itself, the cyclin-dependent kinase, has to be phosphorylated at one or more sites and dephosphorylated at others. So the phosphorylation will be carried out by some other kinases, okay? And the dephosphorylation will be carried out by some phosphatases, all right? So this has to happen before it can become enzymatically active, right? And the removal of the inhibitory phosphate groups by a specific protein phosphatase is the step which will activate the kinase at the end of interphase, right? And once it is activated, it's like a positive feedback mechanism. A cyclin CDK complex, it can activate more of them, more cyclin CDK complexes by a positive feedback type of mechanism. And this positive feedback type of mechanism is what causes this sudden explosive increases in MPF kinase activity, which will drive the cell abruptly into M phase. Okay. So cyclin is not the entire story. There is more to it, right? Now, here in this chapter, right, we have basically focused attention on the cyclin-dependent kinase complex that constitutes the M phase promoting factor, right? But there are many stages in the cell cycle, okay, and each one has got its own cyclin-dependent kinase and its own cyclins, right? So there are many varieties of cyclins, and in most eukaryotes, many varieties of cyclin-dependent kinases which are involved in the cell cycle control. For example, S phase cyclins, again, S is for synthesis, okay, DNA replication, right? So S phase cyclins will trigger the entry into S phase, while G1 cyclins will act earlier in G1. All right? Again, G1 was gap 1. Gap 1, the gap between M phase, all right, and the DNA replication phase. These latter, these G1 cyclins, they will bind to cyclin-dependent kinase molecules to help initiate the formation and activation of the S phase cyclin CDK complexes and thereby drive the cell towards S phase. Note that the concentration of each type of cyclin is going to rise and then fall sharply at a specific time in the cell cycle to control the timing of a particular stage, right? So this is like the timing, the clock that you have in a computer, right? So as before, the sudden fall in the cyclin concentration is the result of cyclin degradation by the ubiquitin pathway. Now, as we have already seen, the cell cycle control system triggers the events of the cell cycle in a specific order, right? You're going to have mitosis, then you're going to have growth, then DNA replication, more growth, again mitosis, and so on. Okay, it has to happen in a specific order. So if one of the steps is delayed, the control system of necessity must delay the activation of the following steps so that the sequence is maintained. And this is accomplished by the action of what are called molecular breaks that can stop the cell cycle at specific checkpoints, right? For most cases, the detailed molecular mechanisms involved are not well understood. And again, the caveat here is that by now maybe they are well understood, because even the other day somebody got a Nobel Prize for figuring out the circuit in the GPS in the brain, okay? So people are working on these things, but from the time that I made the notes, they were not well understood. Maybe some of them are beginning to be understood. I don't know. So however, in some cases, we do understand that, and we know that specific cyclin-dependent kinase inhibitor proteins come into play. Proteins which go and block the activity of the cyclin-dependent kinases, they come into play. And there are a few good examples of that. Now, one of the best understood checkpoints stops the cell cycle in the G1 phase if the DNA is damaged, right? This makes sense because if you have damaged DNA, if the cell replicates DNA, right, you're going to be propagating damaged DNA, right? So that needs to be repaired, right? So there is a molecular break that does that. So this helps to ensure that the cell does not replicate damaged DNA. Now, if there is DNA damage in the cell, this causes an increase in both the concentration and activity of a gene regulatory protein which is called P53, right? This is very, very, I mean, you'll hear about it all the time in the context of cancer, right? And P is basically protein 53 means it's a protein with a molecular weight of 53,000 atomic mass units, so 53,000 Daltons, right? Now when P53 is activated, P53 is the transcription factor, right? Remember, we were looking at eukaryotic transcription. You had that thing that looked like a mouse, a lot of things piling on it to get it going, okay? And those were called transcription factors. P53 is the transcription factor. So when activated, P53 stimulates the transcription of a gene which encodes a cyclin-dependent kinase inhibitory protein called P21, right? Or again, P21 means protein with a molecular weight of 21,000 units, right? So when P53 is activated, it is going to stimulate the transcription of P21. So the P21 protein will form, right? And when the concentration of P21 increases, the P21 protein will bind to the S-phase cyclin CDK complexes responsible for driving the cell into S-phase, and it will block their action, right? So it will delay entry into S-phase, right? It'll bind this the cyclin CDK complexes that are responsible for getting things into S-phase, right? Block their action and delay it until the DNA can be repaired. Right? That is one activity of the P53 gene. So the arrest of the cell cycle in G1 is going to allow the cell time to repair the damaged DNA before replicating it. Now, if for somebody the P53 is missing, right, or you inherited defective versions of that from your parents, right, then you are going to basically replicate damaged DNA, right? And you will be very prone to cancer, right? You'll probably get it at a much earlier age than most people, So if P53 is missing or defective, the unrestrained replication of damaged DNA leads to a high rate of mutation, right? Every time you're dividing, and there's no way to stop that. You step out into the sun, some of your T's got messed up, okay? And if you smoke, some of it causes more DNA damage, okay? All kinds of things happen, okay? If you smoke, maybe the methyl group gets on top of the DNA, okay? So you're going to have G with some methyl groups on it, okay? And that is going to fool the DNA replication machinery, okay? So that's why all these things, these are mutagenic, and they're also carcinogenic, okay? Because they will cause changes in your DNA. So in fact, mutations in the P53 gene that permit cells with damaged DNA to proliferate play an important part in the development of many human cancers, right? Because you have lost the ability to arrest the cell cycle in the G1 phase, right, and prevent the damaged DNA from being replicated. Yeah, go ahead. Yeah, G1 phase is after mitosis and before the S phase. Yeah. No. See, what is going to happen is that, let's say the DNA has been damaged, right? So normally the cell would go ahead and divide again, replicate the damaged DNA and make 2 copies of itself. But here the cell cycle will not continue to the S phase until the repair mechanisms, right, have gone ahead and repaired the DNA, right? Let's say on one of the strands there is DNA damage, right? The cell has mechanisms for cutting that portion out, looking at the complementary copy, right? Making a copy, stitching it back with DNA ligase, and then it's repaired. Okay? Then it's ready to be replicated. Yeah. No, G2 is after DNA replication. This is G1. It will arrest in G1. Yeah. And in fact, the biologists refer to this as G1 arrest. Okay. So. No. It's not after they separate. See, the thing is, it might divide after a long time. Okay. Again, all right. Yeah, the cycle is divide, right? Then for some time, grow, right? And then again divide, okay? It may happen at different rates, but the thing is, if there is DNA damage, okay, you should not go and replicate DNA that has been damaged, okay, you want to repair that, okay. So if you have a cell, right, and it's going to go and let's say divide, okay, you want to arrest the cell cycle in G1 before it gets to DNA replication, right, regardless of whether it has just immediately divided or not, okay, whether it might have divided 2 years ago, okay, that's fine, but if there is damaged DNA before it divides the next time, right, the DNA needs to be repaired, yeah. Or even if from the previous round of cell division, okay, there is a lot of DNA that is messed up, right? Then also that needs to be repaired, you know. Okay, because the problem is if you go and if the DNA damage happens to be in some place where it's not that important, you're probably okay. But if that DNA damage turns on a cancer-causing gene, okay, then that's going to be a problem, right? It will accelerate cell division like crazy, you know, and you get a tumor, cancer, and you know. Malignancy and you know, metastasis and so on. In fact, today I will finish with the discussion of cancer. Okay, so without being a biologist, okay, at least you'll have some understanding, a simplified version of what happens in cancer. I mean, we have alluded to that a little bit in the previous classes, but I think today we'll be able to wrap it up. Yes, sir.
So is there any way that P53 has to be like replicated?
Yeah.
Because it is just like...
No, P53, see, P53 is a gene, right? When the cell divides, right, the entire genome has to be replicated, right? I mean, the DNA has to be replicated. So P53 also will get replicated, yeah. Yeah.
And the cell doesn't have any mechanism to repair the gene itself?
No, the cell has mechanisms to do repair, yeah.
I mean, when P53 is like defective, the cell doesn't have the mechanism to repair.
It does, but you know, not the mechanism. See, the cell has mechanisms to repair. Oh, you mean if you inherited defective P53 from your parents? Is that what you're talking about, or what?
I mean, in general, when
No. If P53 is acquiring some defect or something, okay, it's not an inherited defect. The cell will repair. Okay, it will repair, but as you get older, the repair mechanisms slow down. Okay. That's a fact of life. Okay, you just don't look old. You know, your repair mechanisms also are slowing down. You know, so in fact, you know, like you guys are not at that stage. But this morning on my way to work, it was pretty funny because the DJ was saying something about, you know, people turning like 29, and as soon as you cross into 30, right, then you feel really bad. Okay, like let's say 19 to 20, 29 to 30, right, and so on. Okay, 39 to 40, and then the guy goes and says, yeah, but by the time you get to 60, it doesn't matter anymore, you know. That time you wake up in the morning, right, and you're trying to figure out if all the parts are working or not, okay. You went to bed with X number of parts working, morning it can become X plus 1, you know. So even the DNA repair mechanisms, they slow down with age, okay. Right, and of course, exposure to excessive sunlight and things like that, it can cause, you know, extensive DNA damage. That's why they talk about, you know, getting skin cancer and, you know, to avoid the sun and things like that, yeah. But this one, the P53, the mutations that I'm talking about, these are, some of them are inherited, okay. I mean, like if your parents gave you defective P53, right, then you're in trouble, okay. Then there's another gene that I will talk about, all right. It's called the retinoblastoma gene, all right. Typically kids that have that gene, right, they die by the time they're 2 years old, okay. They develop tumors in the eyes, all right. That's why it's called retinoblastoma gene. But I'll talk about that towards the end of the next chapter. All right, any other questions? Now, cells can also dismantle their control system and withdraw from the cell cycle altogether. Okay, this business of, you know, grow, divide, they can put an end to that, right? So in the human body, for example, the nerve cells have to persist for a lifetime without dividing, right? That's the reason that if you break your spinal cord or something like that, you know, then you're paralyzed, right? It's not going to repair, right? So these nerve cells, they enter a modified G1 state, which is called G0, in which the cell cycle control system is partly dismantled, in that many of the cyclin-dependent kinases and cyclins disappear, right? So it's not cycling anymore, you know, it's just stuck in that state, right? So as a result, the nerve cells remain permanently in the G0 state, right? Now, mammalian cells will proliferate only if they are stimulated to do so by signals from other cells. If they are deprived of such signals, right? It's like if your neighbors don't tell you, don't kind of cheer you on, you know, you just drop down dead, okay? So that's what's happening to these cells, okay? So for mammalian cells. So if deprived of such signals, the cell cycle arrests at a G1 checkpoint and it'll enter the G0 state, right? So it'll just get stuck over there, right? And there is also something called the programmed cell death that we will get to in a few minutes, right? Now, unicellular organisms such as bacteria and yeast, so right, they have to think only about themselves. Okay, it's not a multicellular organism, so they will grow and divide as fast as they can, and their rate of proliferation really depends on the availability of nutrients in the environment. For an animal cell to proliferate, nutrients are not enough. It must also receive stimulating signals from other cells, usually its neighbors. In other words, in multicellular organisms, cell division is under very tight control, right? An important example of a break that normally holds cell proliferation in check is what is called the retinoblastoma protein, right? The retinoblastoma protein, or RB protein, which is abundant in the nucleus of all vertebrate cells, will bind to particular gene regulatory proteins, preventing them from stimulating the transcription of genes that are required for cell proliferation, right? The cell might need certain proteins in order to multiply, okay? So this retinoblastoma protein will act as a brake on that, okay? Now, in response, so if there are external signals that are telling to the cell to divide, okay, like some growth factor comes and binds the cell membrane, the cell needs to divide, all right? In response to that, it'll let go of that brake, all right? And then the cell can divide, right? So extracellular signals such as growth factors that stimulate cell proliferation will lead to the activation of the G1 cyclin CDK complexes mentioned earlier. These will phosphorylate the retinoblastoma protein, altering its conformation. Remember, we said before, that's why I emphasized that in chapter 4. When we did proteins, okay, if you put a phosphate group on a protein, you put in some negative charges, you change its shape, okay. You can change its shape from active to inactive and vice versa, right? So these will phosphorylate the retinoblastoma protein, altering its conformation slightly so that it releases its bound gene regulatory proteins, which are then free to activate the genes that are required for cell proliferation to proceed. The stimulating signals which act to override the brakes on cell proliferation are mostly protein growth factors, okay. These are signals that will come and bind the cell membrane, okay, and there is usually the receptor protein, right, that is conveying the growth factor signaling from outside to the interior of the cell. That will be like an alpha helix that goes through the cell membrane, okay. And these are actually called receptor tyrosine kinases because they usually contain the amino acid tyrosine, right, and it's a receptor that is bound on the cell membrane. So one example of a protein growth factor is the so-called platelet-derived growth factor, right, PDGF, right. So when blood clots in a wound, for example, the blood plates that are incorporated in the clot are triggered to release this platelet-derived growth factor. That is going to bind to the receptor tyrosine kinases, okay. Again, I mean, it's basically a receptor on the cell membrane, okay. And it's a kinase because it is going to cause some phosphorylation reactions inside, right. Tyrosine because the predominant amino acid in that alpha helix is tyrosine, right? So it'll bind over there and that'll stimulate the surviving cells at the wound site to divide, right? And that's how the wound heals because as I've been saying time and again, okay, there is a dynamic equilibrium between cell proliferation and cell death, right? The cells on your skin are replaced once every 10 days, in your gut once every 3 days, okay? So if this dynamic equilibrium is disturbed, then you're in trouble. Normally you don't see anything because the rate of growth is approximately the same as the rate of death, right? But if you have a cut or a wound, for that to heal, you need growth to be greatly turned on, okay? You also need growth to be greatly turned on if it's a child that is developing in the womb, right? Because from a single cell, that has to be an entire individual in 9 months, okay? So if it is just doing, if the equilibrium is not heavily biased in the direction of proliferation, that growth is not going to take place. But for adults like us, there has to be an equilibrium, right? And so if you have a cut or something, in order for that to heal, again, that genes are involved, okay? If you don't have the right genes and the right stuff, you can go to the best doctor in the world, they're not going to be able to make your wound heal, right? So another example of a protein growth factor is the hepatocyte growth factor, right? So that actually stimulates the liver cells to divide. So in this case, if part of the liver is lost through surgery or acute injury, cells in the liver and elsewhere will produce this protein called hepatocyte growth factor, which will help to stimulate the surviving liver cells to proliferate. So like if somebody goes ahead and cuts off, let's say 90% of the liver, the individual can still live because the liver cells can be stimulated to proliferate. Whereas the same is not true for the nerve cells because the nerve cells do not divide, right? Now, even in the presence of these growth factors, normal animal cells do not keep on dividing indefinitely in culture, right? Even cell types which maintain the ability to divide throughout the lifetime of the animal will stop dividing after a limited number of divisions. And this may have something to do with telomere shortening. Remember, we talked about centromeres, telomeres, replication origin, right? The telomeres are the sequences that are needed at the end of linear chromosomes so that that portion can be replicated properly. There is an enzyme called telomerase, okay, which will add copies of that portion just to keep the chromosome long enough, right? And but still it's supposed to shorten with each cell division, right? Now, if telomerase becomes hyperactive, right, then even after a large number of cell divisions, right, the chromosome is still being used, okay? It's not discarded, and that can cause cancer too, okay? I mean, that has been seen in a number of cancers, right? Telomerase hyperactivity can contribute to cancers. Right? Now, so the cells will divide a certain number of times, right? After that, they will stop. For example, fibroblasts, which is a class of cells from which connective tissue is derived, taken from a human fetus, they will stop dividing after 80 rounds of cell division, right? In culture, you're growing them. While fibroblasts that are taken from a 40-year-old adult, they stop dividing after 40 rounds of cell division. So there is a difference between, being like a small child and being an adult, okay? The number of times that your cells have left to divide, okay, it also goes down, right? So this phenomenon is known as cell senescence, okay? So that it's not just you that's growing old, it's even your cells are growing old, okay? Another example is fibroblasts taken from a mouse embryo, on the other hand, will halt their proliferation after only about 30 divisions in culture. So the human one is doing 80 rounds of division, right? And this may partly explain the difference in size between mice and humans, okay? Because the human one divides many, many more times, right? Now, the mechanisms that halt the cell cycle in either developing or aging cells are not clearly understood at the present time. And again, the caveat is some of it may be understood, because there have been people getting Nobel Prizes and all that. I haven't tracked them, but somebody might have found out some of these things. Although the accumulation of cyclin-dependent kinase inhibitor proteins and the loss of cyclin-dependent kinases are likely to be involved. Now, another aspect, in addition to proliferation, there is also this aspect of programmed cell death, right? Animal cells require signals from other cells to avoid programmed cell death, right? So it's not just survival signals, So if they don't get these survival factors or these survival signals, then they will activate a suicidal procedure and die, okay? So if deprived of such survival factors, the cells activate an intracellular suicide program and die by a process which is called programmed cell death or apoptosis. Right. So this helps ensure that cells survive only when and where they are needed. And the amount of programmed cell death that occurs in both developing and adult tissues is astonishing, right? For example, in the developing vertebrate system, where you basically have the nerve cells, okay, and there has to be an interconnection, right? More than half of the nerve cells normally die soon after they are formed, okay? That's because this has to be like an electrical network, okay? So things have to be connected together. If there's stuff left over, it shouldn't be hanging around, So that is going to undergo programmed cell death. Now, in a healthy adult human, billions of cells die in the intestine every hour, okay? So by the time you finish this class, billions of cells have died, okay, in your intestine already. Okay? Now, a natural question arises, what is the purpose served by this huge amount of cell death? In some cases, the answers are clear. For example, the hands and feet, right? It's initially something like this. It's a block, okay? But then you have the fingers that have to be carved out. And that is achieved by programmed cell death, right, in between, right? Same way, the way a tadpole gets rid of its tail when it becomes an adult, okay? That is programmed cell death, okay? Nobody takes a knife and just chops off the tail, It's basically programmed cell death, right? Now, in the case of the developing vertebrate nervous system, apoptosis is used to match the number of nerve cells to the number of target cells that require innervation. That's what I was talking about. It's like a circuit, okay? Neural circuit, right? So extra cells need to be gone, right? In yet another case, an enlarged liver can be returned to normal size via apoptosis, right? So and again, I'm not sure about this, but when people have enlarged livers, all right, they give them medication, okay? Maybe those induce apoptosis in the liver, okay? I'm not sure about that, okay? I haven't looked at the literature because nobody goes and chops off a portion of the liver, all right? So it's probably a programmed cell death or apoptosis that does it. Now, we next examine how apoptosis uniquely differs from other kinds of cell deaths, right? The cells that die as a result of acute injury typically swell and burst and spill their contents all over their neighbors, a process called necrosis. That's when you get an infection or something. If you have an infection and cells are dying at that site, that is necrosis, okay? You don't want to even look at it, okay? But on your skin and all, cells are dying all the time, okay? But nobody cares, all right? So, cells that die by necrosis, they cause a potentially damaging inflammatory response. By contrast, a cell which undergoes apoptosis, it dies very neatly without damaging its neighbors. Right? So the cell shrinks and condenses. The cytoskeleton is the thing that maintains the structure inside the cell. Right? The cytoskeleton collapses, the nuclear envelopes disassembles, and the nuclear DNA breaks up into fragments. Right? Most importantly, the cell surface is altered, displaying properties that cause the dying cell to be cleaned up immediately, either by its neighbors or a macrophage, which is like a trash cleaner, before there is any leakage of its contents. Okay, so in apoptosis, the cell dies a very peaceful death, right? Whereas if you're looking at necrosis, you know, it might be somebody with a dirty bomb that's blowing himself up and killing a lot of other people in the process, you know. So the analogy will be similar, but apoptosis is just, you know, internally, you know, the machinery will get dismantled, and then it'll get even cleaned up by the cleaning machinery of the cell. So that's the difference between apoptosis and necrosis. Yeah. Yeah. So if you have a tooth infection or something like that, you know, there could be necrotic tissue in that. What? Cell what? I didn't understand. Yeah, necrosis is not a nice type of cell death, you know. That's the one that we normally talk about, you know. Like, I mean, nobody talks about, if you're not a biology major or, you know, taking this course, you know, you won't worry about apoptosis, okay. Cell death is basically the infection and the pus and all that you see, okay. So, I mean, that'll be necrotic tissue, okay. So, I mean, it's not nice to look at, right? But apoptosis is very clean, right? And apoptosis has to happen. So, there are two ways you could get cancer, okay. One is when there is excessive cell proliferation. The other one is that the apoptosis is not happening. Okay, so the cells are staying on longer than they should. Then they'll have more damaged DNA, right? So that could also, so cell death is also a good thing, right? Now the machinery that is responsible for this kind of controls cell suicide seems to be similar in all animal cells. It involves a family of proteases. Remember, we talked about enzymes which break up proteins are called proteases. Okay, so there's a family of proteases. And in fact, they are called caspases, okay, cysteine aspartate protease, okay. So I mean, they have the cysteine and aspartate as their amino acids, okay. They're called caspases. So it involves a family of proteases which are themselves activated by proteolytic cleavage in response to signals that induce programmed cell death. The activated suicide proteases cleave and thereby activate other members of the family, resulting in an amplifying proteolytic cascade, which is often referred to as an apoptotic cascade, right? So something gets the reaction started, it's like a chain reaction, and then everything starts falling apart, okay, but very, very neatly, right? Now the activated proteases then cleave other proteins in the cell, killing the cell quickly and very neatly. Okay, any questions so far? Or you're ready to jump into cancer, right? So that's one of the aspects of our lab, of our center. One aspect is the agri-life research, the other aspect is cancer. Okay, so this chapter is important for you. And you know, a lot of the work that has gone on here in genomic signal processing is motivated by cancer. So what are cancers? Cancer is like a breakdown of the cell cycle control system, right? So if you're talking about a child develop in a womb, okay, that's like stability, right? Whereas cancer is instability, okay, everything is messed up, right? So what are cancers? Cancers are the product of mutations that will set cells free from the usual controls on cell proliferation and survival. A cell in the body, a single cell in the body mutates through a series of chance events, and it acquires the ability to proliferate without the normal restraints. Because I talked about all these survival signals, okay, and they will die if they don't get signals from other cells, okay. But the cancer cells become deaf to those signals. So the progeny of the cancer cell will inherit these mutations and will give rise to a tumor that can grow without limit, right? And there are two kinds of genes that can cause cancer. One would be what are called oncogenes, right? This is like the accelerator of a car, right? If your accelerator is stuck, then you're in trouble, okay? But if your one accelerator is stuck, you're in trouble. That's an example of an oncogene. If both your brakes have failed, then also you're in trouble, right? That's a tumor suppressor gene. So oncogenes which turn on cell division and tumor suppressor genes which function as brakes on cell division play an important role in causing cancer. An example of an oncogene is the gene that is produced from the gene for the platelet-derived growth factor, right? A few slides ago, I talked about the platelet-derived growth factor, right? If you have a cut or a wound, platelets at the site will release platelet-derived growth factor, which in turn will go and bind The receptor tyrosine kinases are the surviving cells and stimulate them to divide. Okay, but you don't want this to happen. You don't want platelet-derived growth factor to start acting just by itself. You know, there's no cut or anything like that. If that happens, so this platelet-derived growth factor is called a proto-oncogene. Right? That means before it has become an oncogene. If it mutates, okay, it can become an oncogene, right? And that's when it is going to turn on cell proliferation even when the stimulus is missing, right? And then you'll get a tumor, right? So here, the normal platelet-derived growth factor gene is called a proto-oncogene. That means before it has become an oncogene. And the corresponding cancer-causing gene is called an oncogene, right? While the proto-oncogene is going to stimulate cell proliferation when more cells are needed, like for example, when you have a cut, right? This particular one will stimulate the production of more cells. The oncogene is going to cause cell proliferation even when no new cells are needed, right? So this is an example of an oncogene, right? And there are many others. You know, RAS is an oncogene, right? The RAS protein that's coded by an oncogene. An example of a tumor suppressor gene is P53 is a tumor suppressor, okay? Because if you have both copies mutated, then you're in trouble, okay? It's a tumor suppressor because it works like a brake on cell division, right? Now, another example of a tumor suppressor gene is the retinoblastoma gene, whose product normally acts as a brake on cell division. We talked about this a short while ago, right? Now, when this gene is mutated, the braking action may no longer be present. So normally what is going to happen is that the braking action is there, but to get rid of the braking action, you need the growth factors, right? But if this retinoblastoma gene is mutated, maybe even without the growth factors, the braking action is removed, right? And then the cell can proliferate, right? So it could lead to excessive cell proliferation. In fact, you know, people who have this defective retinoblastoma gene, right? Both the copies are defective. Those are the kids that by the time they're 2 years old, they have these huge tumors in their eyes and they die of retinoblastoma, right? So in a normal diploid cell, that means 2 copies of each gene or each chromosome, right? So since a proto-oncogene accelerates cell division when called upon to do so, only 1 copy of a proto-oncogene needs to be mutated to an oncogene before excessive cell proliferation will and possibly cancer can result. On the other hand, if you have a tumor suppressor gene, it's like a brake on cell division. So both the brakes have to fail, right? So a tumor suppressor gene is associated with braking action on cell division, and so both copies have to be mutated before a loss of function, possibly leading to cancer, can occur, right? So if one parent gave you a defective P53, you probably still have some way to go, you know, because before the other one gets messed up. Now, in almost all adult tissues, cells are continually dying and being replaced. Through all the hurly-burly of cell replacement and tissue renewal, the architecture of the tissue must be maintained, right? We don't keep on changing our appearance every day, you know, just because a few cells died or something like that, okay? Because of apoptosis. So we have some kind of a dynamic equilibrium between cell proliferation and apoptosis. Now, 3 main factors contribute to make this possible. Number 1, cell communication. The cells communicate with each other, right? Believe me, cells learn to communicate a lot before we put in all these wireless systems and, you know, cell phones and all that, okay? So this ensures, cell communication ensures that new cells are produced only when and where they are needed, okay? Then there is also selective cell-to-cell adhesion. The selectivity of adhesion prevents the different cell types in a tissue from becoming chaotically mixed, okay? Like if it's a liver cell, it'll probably, you know, stick with the liver cells, okay? It's not going to get mixed up with other cells. And then also there is cell memory. Cells autonomously preserve their distinctive character and they will pass it on to their progeny, right? And this preserves the diversity of cell types in the tissue. Now, different tissues in the body are renewed at different rates. For example, the nerve cells in the body never divide, while the liver cells divide about once a year. The cells lining the inside of the intestine, on the other hand, are turned over once about every 3 days, right? So many of the differentiated cells that need continual replacement are themselves unable to divide, okay? Because they're not stem cells anymore. So more of such cells are generated from a stock of precursor cells, which are called stem cells, which are retained in the corresponding tissues along with the differentiated cells. So this is like whenever more new cells are needed, then they will be produced from the stem cells, okay? And then these will be differentiated, and some of them will be stem cells, all right, for future use, okay? Some cells are being used right now, some others, basically the reserve for future use, for generating cells for the future. So stem cells can divide without limit. Some of the progeny of the stem cells will remain stem cells, while others will embark on a course leading irreversibly to terminal differentiation, so to producing the cells and tissues that are needed at that point in time. Now, Cancer cells are defined by two heritable properties. That means they can pass it on to their offspring. They and their progeny will reproduce in defiance of the normal social constraints, right? And they will also invade and colonize territories normally reserved for other cells. Cells which have only property 1, that means it is just multiplying, okay, uncontrollably. That will result in a tumor, because cell growth is more than cell death, right? You'll get a tumor, but it is benign. While cells which have got properties 1 and 2 will result in a malignant tumor, and that is cancer. So that's why many times so if you have any tumor, okay, it's better to take it out, because it has the potential to become a cancer, okay. Because tumor means cell division has been turned on, okay. You're multiplying very fast. Every time you multiply, there is possibility of DNA damage, okay. And it's only a question of time before the oncogenes get turned on or the tumor suppressor genes get turned off. So it's a good idea if anybody has tumors, to go and get them taken out. That's why they go ahead and biopsy it. If it hasn't become malignant, then you're fine. It's as good as before, right? If it has become malignant, then you have to worry about whether it's spread or not, right? Now, malignant tumor cells can break loose from the primary tumor, enter the bloodstream or lymphatic vessels. And once that happens, they can go anywhere in the body, right? And they can form secondary tumors, or what are called metastases, at other sites in the body, right? And in everyday language, this metastases is what is referred to as the spreading of cancer. Somebody got breast cancer, now they got lung, or brain cancer, and so on, okay? Because it starts spreading. Now, it is important to note that cancer is a consequence of mutation and natural selection within the population of cells that form the body. All right, so the principle of survival of the fittest works, okay, because the cancer cell is the fittest one, okay. It's only looking out for itself, all right. So indeed, the mutations in cancer cells give them a competitive advantage over their normal neighbors. Unfortunately, it is this very fact that leads to disaster for the multicellular organism, since the controls on cell proliferation and survival, which are at the heart of maintaining tissue architecture, are disrupted, right. About 10 to the power of 16 cell divisions take place, and I don't know how they came up with the math, okay, but 10 to the power of 60, that is a big number, right? Cell divisions take place in the human body over the lifetime of an individual. And based on this, they can do the math, and you guys should do it, because you're the people with the quantitative skills. You can do the math and show that every single gene in the genome is likely to have undergone mutations on about 10 to the power of 10 separate occasions, right? In any individual human being, right? So there's no point in worrying about this. The mutations are happening, okay? But hopefully the, repair mechanisms will kick in, right? Now, typically at least 5 or 6 independent mutations must occur in one cell to make it cancerous, right? So this is because the body of a multicellular organism has got different levels of protection against cancer. For example, to become cancerous, an epithelial stem cell, that means a cell, let's say, on your skin or lining the gut, okay, must undergo changes that not only enable it to divide more frequently than it should, but also let its progeny escape being sloughed off in the normal way from the exposed surface of the epithelium. Remember, the cells on your skin are replaced once every 10 days. The cells lining your gut, once every 3 days, okay? So even if the cell is trying to become cancerous, okay, in the normal procedure, it should be gone, okay? In 10 days, it's gone, right? So it needs to do something more than that, in order to develop full-blown cancer, right? Then they also have to be able to displace their normal neighbors, right? And they also have to attract a blood supply sufficient to nourish tumor growth. Because for a tumor to grow, it is going to need nutrients, right? So it has to develop that. That's why they talk about the generation of new blood vessels in cancer, which they call angiogenesis, okay? Because the tumor is going to, give birth to a blood vessel so that it can draw nutrients to it. Thus, the mutations that give rise to cancer accumulate over a long period of time. And that is why cancer is typically a disease of old age, right? Assuming that, your genes were not messed up when you were born, right? Probably you're okay, But if there's a child that has inherited defective copies, then they will not last very long. So occasionally, however, individuals are encountered who have inherited a germline mutation in a tumor suppressor gene or an oncogene, right? They got it from their parents. For these people, unfortunately, the number of mutations that is required is less, right, to get cancer, right? And the disease occurs more frequently and at an earlier age. And the families that carry such mutations are therefore prone to cancer. Okay, I think I'm done with cancer. So any questions? So did you get some understanding of cancer? But of course, it's a lot more complicated than this, because see, cancer is basically the ways in which the cell cycle can break down, okay? And if you've studied control systems, okay, you would know that if you're talking about a linear time invariant system, there is only one way in which it can be stable. All the poles have to be in the left half plane, okay? But the number of ways in which it can be unstable is, infinite, okay? You could have one pole in the right half plane, two in the right half plane, so on, three on the j omega axis, okay? So, in cancer, because it's a signaling breakdown, right? And there's lots of redundancy. So typically the challenge, there are a lot of challenges associated with cancer treatment, okay? First of all, it has to be personalized. No two cancers are the same, okay? Because it's a breakdown. Then the second thing is that even the cancer tumor, okay, it is going to be heterogeneous, all right? The different cells in the tumor will have different breakdowns, okay? So if you give it some drug that is targeting one clone of cells, okay, it's not going to cure the others, all right? So you see typically, people respond initially to the therapy, right? And then the cancer figures out a way, to get around it, right? And like one of my biologist collaborators says, the cancer drugs are only about 50, 60 years old, okay? The cancer is a very intelligent person, okay, because it's been around for millions of years, so you're trying to fool it with the drugs, it'll find a way around it, okay? So it is challenging. That's why there is interest in what is called combination therapy, where you're going to use a number of different drugs, okay, so that if this one doesn't get it, that one will get it, and so on, okay. But again, with sufficient time, it can probably figure out a way to get around it. In fact, the only cancers for which there have been remarkable success, all right, these are the leukemias, and for these, the breakdowns usually occur in a very, very predictable fashion, right, at one particular location. So they know what drug to use, but for most of the cancers, that's not the case, right? And the current state of the art in cancer treatment is the following, right? If somebody is diagnosed with cancer, they have a bunch of, anti-cancer drugs, like for example, that colchicine that I told you, okay? So it'll go try to kill the cancer cells, all right? So they'll give you, some of those drugs, all right? One after the other, they'll try, either one of those works or you die, okay? So that's the state of the art. It's getting a little bit better, but I'm saying that is the state of the art. And that's the reason why all of you guys are here. Hopefully you're going to generate the breakthroughs in therapy. So because you need people with quantitative skills. If you can, do molecular profiling of the tumor, hopefully you can come up, with a better strategy, right? Which drugs to use and so on, okay? So the next chapter is going to be on expression microarrays, all right? And I'm not going to start the chapter today, all right? I'm just going to mention what is involved here. And again, the molecular biology portion is over, okay? So you can start studying for exam number 2 right now, okay? And you have like 3 weeks to absorb. Is it 3 weeks or a little bit more, I think, right? It's Monday. Yeah, it's November 3rd. So you can start studying for that, because there will not be any more biology to memorize, okay? So we have finished that part, right? Now, what are microarrays? See, microarray is like a gene chip, right? Like, see, in the past, people would focus, let's say, on one particular gene, okay? So how will you go and detect a gene? If you have its complement, right? The complement of, because we know that DNA always forms the double helix, okay? So if you're looking for a particular gene, let's say the hemoglobin gene, you will take the complement of that gene, right? And see if something from your sample hybridizes to it or not, okay? That's how you would look for it. In the case of microarrays, you're doing that for thousands of genes at the same time, okay? And really, these expression microarrays, you're trying to figure out the activity of the genes, okay? How will you figure out activity? That'll depend on the RNA that is produced in a particular tissue, okay? So you want to look at the RNA, because the gene is already there, okay? It's there in the DNA. But the gene that is expressed as messenger RNA, right? You take that. From the messenger RNA, right? You can construct what is called the complementary DNA, right? cDNA or complementary DNA. And then you will see, let's say you want to differentiate between two samples, right? Is this, let's say, normal versus, let's say, cancerous, okay? And you want to figure out which are the genes that are differentially expressed, right? Then what you would do is you would create the complements of the genes that you're interested in as probes, okay, on a glass slide, right? And then get these two samples, all right, labeled with different colors, all right, to hybridize in there, shine a light, get an image, okay, and then look at it, okay? And that's where you will need image processing, signal processing, and so on, okay? So EE is coming into the picture, right? This is not a course on molecular biology, but this stuff I had to cover, right? My apologies, it's been very fast, but most people did well on the exam, so I think it's fine, okay? Because most of you didn't know any biology, so you learned something, and you can learn more stuff from the internet, okay? Even oncogenes, tumor suppressor, and you can understand what's going on, right? So next time I'm going to start with expression microarrays, all right? And, like Mike Bittner, who's at this center, right? Have you met Mike Bittner or no? Yeah, Dr. Doherty and Mike Bittner, they worked on the first paper that did the image analysis of microarrays, right? Analysis of microarray images. This was way back in '97. Okay, so I will cover that paper in the class. First I will just go through the PowerPoint, and then I have some notes on that paper. I will hand that out, and I will cover that. And again, I mean, we are going to have to leave enough time, right, in this course for 17 people to make presentations. Okay, you guys are going to make presentations, so I have to finish all the material, and probably we'll have like 2.5 weeks left, right, when we'll use the class time to make presentations, like maybe 3 people present each day, right? Maybe we start a little bit early, 12:30 to 1:30, or maybe 12:30 to 12:40 to 1:40, right? And then finish it up. So now is probably the time when I mean, you don't have to pick up your paper right now, but as we go along, you should be looking either at our website or somewhere what paper you want to present, okay? Of course, if somebody comes up with a neat idea, you want to write your own paper and present, that's fine too, okay? I mean, you suddenly got excited by learning all this biology. Hey, I have a solution to this problem and I can work it out. That's fine. No penalties for that, okay? So, all right. Any questions?